DNA methylation changes. DNA methylation is the addition of a methyl group to a cytosine base, typically in CpG islands, which are regions of DNA rich in cytosine and guanine. In colorectal cancer, two primary types of aberrant DNA methylation occur. Global hypomethylation. This is a widespread decrease in DNA methylation across the genome. It is a hallmark of many cancers, including colorectal cancer, and often affects repetitive DNA sequences. This can lead to genomic instability and the activation of genes that should be silenced, such as oncogenes. Hypermethylation of gene promoters. In contrast to the global trend, the promoter regions of specific genes, particularly tumor suppressor genes, become excessively methylated. This hypermethylation leads to the silencing of these genes, which would normally control cell growth, DNA repair, and apoptosis, programmed cell death. Histone modifications. DNA is tightly wound around proteins called histones to form chromatin. Histone modifications, like acetylation and methylation, change how tightly the DNA is packed, which in turn controls gene accessibility and expression. Altered chromatin structure. In CRC, an imbalance of enzymes that modify histones can lead to an incorrect chromatin structure. For example, increased histone acetylation in certain regions can loosen the chromatin structure, allowing for the transcription of oncogenes. Conversely, increased histone methylation can compact the chromatin, silencing tumor suppressor genes. This dysregulation alters the balance between cell growth and death.
MicroRNA mRNA patterns. microRNAs are small non-coding RNA molecules that regulate gene expression after transcription. They bind to messenger RNA, mRNA, and prevent it from being translated into a protein, effectively silencing the gene. In CRC, the expression of these mRNAs is often disrupted. Tumor suppressor mRNAs. Some mRNAs act as tumor suppressors by targeting and silencing oncogenes. In CRC, these beneficial mRNAs are often downregulated, meaning their levels are low, which allows cancer-promoting genes to be overexpressed. Oncogenic mRNAs, OncoMRs. Other mRNAs, known as OncoMRs, promote cancer by targeting and silencing tumor suppressor genes. In CRC, these mRNAs are often upregulated, leading to the inactivation of genes that would otherwise control cell growth and death.
Medicine and disease management. Early detection and diagnosis. Epigenetic markers can serve as powerful biomarkers for the early detection of diseases like cancer, even before symptoms appear. For instance, abnormal DNA methylation patterns can signal the presence of a tumor. Personalized medicine. Understanding an individual's unique epigenetic profile allows for the development of personalized treatments. Instead of a one-size-fits-all approach, doctors can tailor therapies based on how a patient's genes are expressed, improving the effectiveness of drugs and reducing side effects. Licensed by Google Novel Therapies. Because epigenetic changes are reversible, they present promising targets for new drugs. Epigenetic therapies, such as epigenome editing tools and drugs that inhibit or activate specific enzymes, are being developed to correct faulty gene expression patterns in diseases like cancer and neurological disorders. Understanding disease mechanisms. Epigenetics helps explain why some individuals with a genetic predisposition for a disease never get sick, while others do. It bridges the gap between genetics and the environment, revealing how factors like diet, stress, and toxins can alter gene expression and contribute to disease development and progression. Agriculture and crop improvement. Improving crop resilience. Epigenetic research is crucial for developing climate-smart crops. By understanding how epigenetic mechanisms help plants adapt to environmental stresses, like drought, heat, and salinity, scientists can engineer crops with enhanced tolerance. Enhanced traits. Epigenetic modifications can influence important agricultural traits, such as yield, flowering time, and disease resistance. By manipulating these epigenetic changes, plant breeders can improve crop productivity and quality. Epibreeding. This is a novel approach that uses epigenetic variation to develop new crop varieties. By identifying and selecting plants with beneficial epigenetic markers, breeders can accelerate the creation of superior crops, leading to more sustainable and productive agricultural systems. Sources.